Hi, my name is Hayden Henry. I am a DOE Sully student and currently a rising junior at University of Washington pursuing a major in biochemistry. At PNNL, I work in the biological science division, specifically the metabolomics group under the mentorship of Tom Metz. Today, we're going to be talking about metabolomics for analyzing the effects of simulated shift work on plasma composition. I wanted to start with kind of outlining why we need to study night shifts. First of all, night shifts are, the night shift population is continuing to grow with a large jump between 2007 and 2017, jumping from 10 to 20 million individuals. Not only that, but night shifts have been associated with a number of diseases, such as cardiovascular disease, type 2 diabetes, cognitive decline, and cancer. In fact, after just 3 days of simulated night shift, we see an increase in biomarkers for cancer. And this study in particular was aimed at identifying early biomarkers of disease in the plasma by examining the metabolites and their compositions. To understand why night shifts might lead to disease, you have to have a little bit of background in circadian biology. And this is just the rhythmic peaks and valleys of physiology and its products and byproducts. And these rhythmic peaks are on a 24.5-hour cycle. So every day we need to do behaviors that reset these clocks. And these behaviors can be things such as light viewing, exercise, eating, and temperature modulation. And the brain then, the central modulator, is going to respond to this by releasing hormones to the rest of the body. And we can see this through something like cortisol, the stress hormone that also kind of promotes movement. We can see at 1 a.m., it's at its lowest point, and then that then rises to 8 a.m. And this makes sense because at 1 a.m., we should be asleep, not moving. And at 8 a.m., it's when the day is beginning and we're getting moving for the day. Now, shift work is going to be a complete disruption of all these behaviors because by shifting when we wake and when we sleep, we are now doing things like eating in the middle of the night when we should be sleeping. We're doing things like light viewing, potentially exercising, and also exposing ourselves to different temperatures. And these shifts can then lead to physiological disruption. We specifically use metabolomics in this study to track the shifts by analyzing the prevalence and structure of metabolites in the sample. And that's what metabolomics is. And metabolites are just any biologically active compound. So we can see here an example of energy metabolism. And we see that glucose, which then becomes pyruvate, enters the mitochondria, which is then going to produce ATP and CO2 that becomes carbonic acid. And these are all metabolites. We have the main compound being consumed, its intermediary, the main product, and the byproduct. And we can measure these with things like mass spectrometry or NMR. This study in particular looked at two groups. One was subjected to night shift and the other was subjected to day shift. The day shift group was awake from 6 a.m. to 10 p.m. and the night shift group was shifted forward 12 hours. So they were awake from 6 p.m. to 10 a.m. And these groups experienced these, were subjected to these conditions for 3 days. And then on the fourth day, they were 24.5-hour samples that were taken every hour and a half from the subjects. WSU did this study and they took a targeted analysis of the samples. And all that means is they had an external reference within the sample. So they also used mass spectrometry. And what this means is with the external reference, they knew what spectral values to look for the 180 compounds specifically they were looking for, and they found 161. PNNL, we took an untargeted standards-free approach, which means we went big. We took everything in the sample, and that's what we were trying to find to identify potential biomarkers for disease. And you have to go big, which means we use mass spectrometry because mass spectrometry allows us to identify a diverse set of compounds within the sample, but also a diverse set of abundances within the sample as well. And traditionally, you have to be able to use a two-dimensional separation, which just means we can identify compounds based on their chemical properties in this case, and their mass properties. And this is done through liquid chromatography, which is separation via chemical properties. In the experiment that we ran, we did a column here that we packed with resin beads. We then washed it with the sample to bind the sample to the beads. And then we washed the sample off with a gradient solvent. So every 20 microliters, we went from a highly organic solvent to a more aqueous solvent. What this allows us to do is select for polarity. Every second or so, that column is going to elute into the mass spectrometer to give us this readout of a mass separations. And this is our mass to charge of each compound. And we can do further validation of compounds within the sample by looking at its MS2 or how it will predictably fragment when bombarded with electrons. However, first I should mention that features are just traditionally have been the liquid chromatography plus the mass spectrometry information. However, at PNNL, we took a three-dimensional approach. We added in this ion mobility spectrometry, which allows us to see structural information. So now on this graph, we have our liquid chromatography. So it's time for elution. And this is the amount of compounds that come out of the liquid chromatography column. This is them going into the ion mobility spectrometer and the mass spectrometer. And we can see we can separate them via their mass to charge ratio and drift time, which is a structural value because when we look at what ion mobility spectrometry is, it's now instead of these ions being eluted into the mass spectrometer, we're now eluting them into this mass spectrometry drift tube. And what this allows us to do is as the ions are pulled through the drift tube, they're going to come in contact with these inert gas particles. And all that means is they're just non-reactive, so something like nitrogen gas or hydrogen gas. And as compounds with the same molecular formula, but different structures fly through this gas, they're going to interact with particles in different ways, and that's going to cause them to slow down. And so when we push them through the drift tube, we can see we can now separate compounds with the same molecular formula by looking at their structure. And this is going to give us a CCS or a collision cross-section, not just a standardized way of looking at it. So now if we look at mass spectrometry in all three dimensions, we see this is what it would look like on the readout. So you can imagine this is us looking at a feature from the front, from the top, and from the side view, kind of. In fact, here's the cloud feature. And the reason this is important is this is the distribution of all the different values we've discussed. So mass to charge ratios, the CCS, and the liquid chromatography values. And what they do is they then take the highest intensity peak and they make that a point, and then they align that point across all samples. And that's our peak picking and alignment across samples. So this gives us a new feature with 3 dimensions of separation that allows for higher confidence in our identification of compounds. Specifically, the flow that was taken in this experiment is Deimos, and that's the software that was used, did the peak picking and alignment. And then what that did is it produced our spreadsheet, mass to charge values, CCS, and also we use MS2. Liquid chromatography is not necessarily standardized across laboratories, but it is a useful separation feature. So what they then do is they use MAME, which takes those average values and compares it to all the predicted and experimental libraries we have available to us. And that produces an annotated data file. And that was approximately 13,000 features. What we then did is we looked at the values from looked at the compounds that WSU identified. Because those are high confidence identifications, we know that those are most likely in the sample. So we picked 43 metabolites, and then we filtered by the 43 metabolites that they identified to give us only 800 features. What I then did was I looked at when the metabolites peaked and if it was a statistically significant peak or a statistically significant shift in peaking. And that by filtering by the statistical significance of the peaking, we got 200 features. What I then did is I examined when WSU found their peaks or found their compounds to be peaking. And then I examined when we found our compounds to be peaking. And as long as they were within one standard deviation provided by WSU, I used that to be the last feature, and that gave us 3 final features. These features correspond to 2 compounds, valine and acetylcarnitine. However, it turned out that valine really wasn't rhythmic. It didn't really have a peak. So even though there was a shift, because there wasn't a peak, we wouldn't really consider it circadian. So this leaves us with just acetylcarnitine. And as we can see here with the graph of acetylcarnitine, there's not only a significant change in the amount of acetylcarnitine present, but there's also a significant shift in the timing of the peak. And to kind of understand what this meant, I ran this through KEGG, which is a metabolic database, and identified that acetylcarnitine is strongly associated or strongly involved with the fatty acid oxidation in the mitochondria. What's the impact of this? Well, if we take a quick look at the acetylcarnitine metabolism, we can see that acetylcarnitine is actually the group that will ferry the acetyl group across the mitochondrial membrane and then provide the acetyl group for CoA, the enzyme responsible for fatty acid oxidation. So elevated acetylcarnitine has been associated with a number of diseases because it could be a sign that there are elevated fatty acids in the blood, and so acetylcarnitine production has to respond to that, or it could be a sign of some sort of mitochondrial issue. In fact, increased abundance could be an early marker of metabolic disease. Not only that, but this was the first use of Deimos and MAME, really in any peak picking, but especially in 3 dimensions where there's not a lot of validation for this 3-dimensional approach in metabolomics. It's been used in proteomics, but metabolomics is kind of an uncharted territory. Thank you to my mentor, Tom Metz, the data scientists, for continually reformatting all the sheets for me, Sam Sakar, and then PNNL for opening the doors for us SULI students. It's been an amazing experience.
Thank you. Well done. Now we're opening it up for question and answer. If you'd like to unmute and ask your question or put it in the chat box. I see we have one in the chat box. Henry, I will go ahead and read that to you. What is retention time and how does that contribute to feature for MS?
Yeah, so I'll go, let's see if I can't go back. Oh, 1 second, have the chat box open. Retention time, that's just going to be, so when I was discussing that 1 second elution, so basically the liquid chromatography sprays directly into either the mass spectrometer or the ion mobility spectrometer. And what retention time is just the time that the compounds come off. So as we slowly go for a gradient, it's like a 10-minute process. So it's a continuous gradient solvent. So over the course of 10 minutes, we have different compounds coming off. And so retention time allows us to basically see, okay, we see the compound coming off at say 5 minutes. So we know this is a more we know this is a more polar compound than non-polar compound because the solution is becoming increasingly polar. So it allows us to separate via polarity. And while it's not necessarily a standardized value across laboratories, what it allows us to do is separate compounds by if compounds do have similar molecular formulas, we can separate by their chemical properties.
Great. He says, got it, thanks. Okay. All righty. Well, if you have any additional questions that you think of, please put it in the chat box. You can directly message Hayden for a response. Again, well done. Next up, we have Daniel Huang. Daniel, you can go ahead and share your screen, unmute yourself, and you have the floor.
Okay. All right. Give me 1 second. All right. Can you see my screen? No? Awesome. All right.
We don't see, you're not in presenter view. Oh, there you go.
Oh,
there you go.
There we go. Okay. All right. Hello. Welcome to my presentation on the integration of the SAGE plasmid delivery system for efficient genetic knockouts. My name is Daniel Huang. I go to UCLA studying psychology and cognitive science, and I am here at PNNL under the synthetic biology division. So as known, climate change has become one of the greatest challenges in the 21st century. Increased temperatures and CO2 emissions bring into question new solutions to combat this rise. Likewise, plant pathogen spread alongside other detrimental effects of human development are also challenges that need to be addressed. We already harness plants for the following uses, such as renewable energy and agriculture, but we plan to use engineering to further promote and access the potential of using plants. So we plan to specifically target plant growth promoting bacteria known as PGPRs. So plant growth promoting bacteria have a symbiotic relationship with plants. They provide valuable tools for plants such as pathogen protection in exchange for habitat and food. So the research goals of the Persistence Control Task Force is as follows. So the first goal is to understand the risks and rewards of genetically modified bacteria deployment. The second is to develop effective containment strategies for the deployment of bacteria. And thirdly is to understand interactions between synthetic bacteria and the natural microbiome. So the group that I was in is focusing on containment strategies. And there's two main containment strategies that we're focusing on: gene reduction and metabolic addiction. So in gene reduction, we're looking to weaken or remove catabolic pathways. Or likewise, we're looking to remove genes that allow for persistence in certain environmental conditions. And secondly is metabolic addiction. This is where we integrate genes in the bacteria that make the bacteria obligate to certain molecules. So ultimately, what we're looking to do is we're looking to restrict the niche of the bacterium and then expand it in a defined manner. So containment strategies, to make these containment strategies, we first must find the function of genes in the bacterial genome, which we did using RBT and seq, or known as random barcode transposon sequencing libraries. So transposons are mobile genetic elements that randomly insert themselves into genomic DNA. For example, if we took 1,000 replicates of bacteria and then we put transposons into every single one of them, we may come out with 1,000 different mutants because the transposons in each of those bacteria may be inserted at different locations. And the barcode attached onto the transposon acts as a specific identifier for that transposon. So what we can do is that after we make the mutants, we can do Illumina sequencing of the barcodes. And what that tells us is that it will read out the barcode. And if we know the barcode location, we know where the insertion site of the transposon is. And what the transposon does is that when it's inserted in the genomic DNA, it will actually interrupt the genetic expression of around the insertion site. So finally, what we can do is we can take these mutants and then do a fitness evaluation in which we measure barcode abundance in a nutrient-rich media, and then we grow them in a selected media and then take the abundance again. And then we can take a gene fitness calculation of that, and then we can find our genes of interest. So for example, let's say we're trying to identify genes that contribute to cold tolerance or heat tolerance in certain bacteria. We would employ the RBT&seq protocol in the bacteria. We grow it in a nutrient-rich media in regular temperature, and then we grow it in a nutrient-rich media in stressed temperature. What we can do is we can compare the frequency of barcodes from the regular temperature versus selected stress temperatures. Let's say we're focusing on one specific barcode. If the frequency of that barcode was lower in the selected stress temperatures, then the fitness frequency change would be negative. And what that means is that if we know what the barcode is, we know that the barcode is specific for a specific mutant. And that mutant, since we already sequenced it, we also know what gene is being interrupted inside that mutant. So what this tells us is that we can actually figure out that the gene that was interrupted may have something to do with stress temperature resilience. So actually, here's an example of RBT&seq results that you may find from an actual run. So on the left is each entry inside the library, and that actually corresponds to a mutant with the transposon interrupting a single gene. And on the top are columns that represent the selected media that we decided to grow the mutants in. So I kind of wanted to focus on one specific condition, which is N-acetylglucosamine, or gluc-NAC. And this is something that can be consumed as a carbon source by the bacteria that we are using. So if we were to focus on this sort of rectangle that I circled here, I have 3 rows, which means there's 3 mutants. And they all have decreased fitness scores when grown only in gluc-NAC. So what does this mean? This kind of means that the genes interrupted in these 3 mutants is related to gluc-NAC metabolism in some manner. And we can do this for all the other conditions and all the other mutants in our run. So the drawback of RBT&seq is such that the process of RBT&seq only targets and tracks the interruption of one gene. But we know that catabolic pathways are often polygenic, such that different processes are controlled by different genes. So in one mutant, if we removed one gene, we'd remove the secondary metabolite, but then the bacteria may just go through a subroute and finish the pathway. So sometimes the impact of gene interruption in RBT&seq is masked because of these alternative pathways. So the next step is to use CRISPR-assisted RBT&seq. And what this means is that we're going to be using a class of the CRISPR protein called CPF1, and then a subclass of that CPF1, which is ddCPF1. So unlike Cas9, which most of you guys are probably familiar with, that excises the gene that it recognizes, ddCPF1 has this DNase remove, which means that when it attaches onto the DNA that the CRISPR RNA recognizes, it will only interrupt transcription, but it won't excite the gene. So what this allows us to do is to do multiplex gene knockdown. So instead of gene 1 being knocked down, we can knock down gene 2, 4, and 5 all at the same time. And this kind of circumvents the issue that we have with RBT&seq, where it's a one-to-one relationship. Now we can have a more comprehensive understanding of different pathways inside the genome. And in order to put CRISPR machinery into the bacteria that we want to test, we're going to use the SAGE system. And there's 2 main parts of the SAGE system. There's the AB cassette, which is in the bacteria of interest, and the AP site, which is on the target plasmid with the cargo that we want to put inside the bacteria. So what happens is that the serine integrase protein will recognize the AB site on the strain and the @P site on the target plasmid, it will recognize both and it will integrate the entire plasmid into the bacteria. So finally, what you get is an integrated @P target plasmid strain, which contains the cargo that we want to put in our bacteria of interest. And this is what SAGE and multi-gene knockdown, this is kind of the pathway that we're taking in order to put CRISPR in the machinery. So we have CRISPR carrying plasmid with its integrase, and we have the CRISPR RNA carrying plasmid with its integrase. And what we can do is at the same time, we can put both cargoes into the genome. And therefore, this becomes a really streamlined process because we can very easily get gene interruption quickly. So my project goal was to merge the CRISPR-I and CRRNA plasmids into one. And there's sort of a mathematical reason for this. So let's say transformation efficiency of uptaking a plasmid in the bacteria could be modeled by this exponential function. And if we're trying to, in order for CRISPR-I to work, it needs both CRISPR-I and CRISPR-RNA. So we can take both. If we take the 4 sets of plasmids to integrate, it's a kind of a low transformation efficiency. But if we're able to half it, then we can increase it by a lot. And when we're trying to make 1,000+ mutant libraries, it becomes really important that we have sort of an extremely efficient way to do so, because these are like week or month-long projects that take a lot of time to complete. So the experimental design went as follows. We took a strain, T fluorescence SU25, J4694, and then we transformed it with plasmids of our choice. We then plated it in selected media, created liquid cultures from that, and then put that in the 96 well plate. After that, we took our well plate and we ran it through a flow cytometer. And the flow cytometer was reading green fluorescence protein from each of these bacteria. And this is sort of the proxy we used to measure whether our CRISPR-I machining was really working or not. And likewise, here are the following conditions that we are testing. So the first condition was a single plasmid system strain. So these strains, we were only integrating one plasmid, which contained both the GDCPF1 and the CRISPR RNA on the same plasmid. And then we had the regular 2 plasmid system, which is what we've been using for the GDCPF1 and the CRRNA sets on different plasmids. And then we have positive and the negative control. And we also layered these conditions on top of another set of conditions, which had the promoter sequences of the GDCPF1 and the CRISPR RNA altered. And this is just sort of a sample photo showing on the left what the positive control looks like. And the positive control has only GDCPF1, but no CRISPR RNA. So you can still see that it's not going to knock down anything because the GDCPF1 won't recognize any sets to land on. And on the right is the single plasmid. So you can see there's a very difference between the amount of green that you can kind of see in the photo. And here are the results. And I think there's like what I circled were 2 really main important results. The first one is at the bottom. So PRS327_PRS323 is the double plasmid system. And PRS319 is the single plasmid version of the double plasmid. So it just contains both cassettes into one. And on the top is the mirrored version, but with different promoter sequences. So what this kind of tells us so far is that 1, the double plasmid systems are outperforming the single plasmid systems in terms of knocking down GFP. So this means that currently our CRISPR RNA and GDCPF1 variants are not synergistic inside a single plasmid. So we believe that this potentially has to do with the homology of the TAC promoters in some of the variants of the CRISPR RNA and GDCPF1. And because we're focusing on the promoters, we're now gonna, in the future, find new promoters using the SAGE promoter library and that either express at the same or higher levels than TAC. So ultimately, even though this initial experiment showed lower knockdown potential, we're still in the future gonna focus on improving the system till such that the single plasmid system is knocking down at the same level as the double, because we really want transformation efficiency as part of the library creation workflow so that we can get more speedy and potent gene discovery. So last but not least, I'd like to acknowledge the following people, especially my mentor, Robert Egbert. He was a great help and he really helped me understand everything I needed for the project. I'd also like to thank everyone on the synthetic bio team for helping me in lab and nurturing me and teaching me everything. Last but not least, I'd like to thank Nancy Rowe, Juan Lopez, Nicole Castelletto Bentley, Brandy Zuger, and all the WTS staff for making the internship as great as it could be.
Thank you, Daniel. That was great work. We're going to open it up for question and answer. If you have a question, please put it in the chat box, or you can go ahead and mute. Daniel, I think we have one in the chat box from Wydong.
Yeah, I got the chat box open so I can read it. So I believe that with viruses, as long as you can edit the CRISPR RNA to read the viral genome, then you could sort of interrupt different portions of the virus gene, and then you could see how the virus would sort of work when certain genes are interrupted.
Great. Any additional questions? All righty. Great work, Daniel.
Great, thank you.
Thanks. Great presentation. Again, if you have additional questions, please put in the chat box and or directly message Daniel. Up next, we have Eve Farrell. Go ahead and share your screen, Eve. You can go ahead and mute yourself and you have the floor. There you go.
Sweet. Thanks, Nicole. Just going to set it up. All right. Hi, everyone. I'm Eve Farrell. I'm a summer Suli intern here at PNNL, and today I'll be presenting on the work that I've been doing under the mentorship of Jim Moran and Chris Krasinski. This is an ongoing project in which we attempt to develop RNA as a taxonomically specific stable isotope biomarker. And we'll be using this to track carbon exchanges between plants and microbial communities in the rhizosphere. So before we get into the project itself, I think it's important to give a bit of background on the rhizosphere specifically. The rhizosphere is a layer of soil just millimeters thick surrounding plant roots, and it's really high in levels of organic carbon, which is released from these nearby plants. Soil microorganisms are then chemotactically attracted to the plant root exudates, which leads to abundant microbial numbers and activities in the spatially constrained. As a consequence, the rhizosphere is replete with essential microbial, geochemical, and plant processes. And this leads to a sort of synergy in the rhizosphere. So for example, the plant root exudates may differ depending on the species of the plant or the environment in which it's in. This shapes the composition of the microbial communities related to them, as well as the Meanwhile, the microbial communities themselves that are associated with these plant roots may confer specific functions to their hosts, which modulates the plant's growth, health, and overall productivity. Therefore, we can see that a comprehensive understanding of this dynamic zone is really useful for studying things like ecosystem stability, and improving our sustainable agricultural practices. This begs the question of how do we measure these interactions? Unfortunately, current techniques such as stable isotope probing, proteomics, lipid biomarkers, and various genomic methods all lack a crucial element of taxonomic specificity. And this just means that this complex web of nutrient exchanges can only be understood fairly broadly, which hinders our trophic and functional understanding of this system. And that's where our project comes in. We intend to quantify the exchange of nutrients, specifically carbon, at a finer taxonomic level by using ribosomal RNA. And this is something that has never been done before. To illuminate how we can eventually use RNA to track carbon in specific microorganisms, it's first important to know that every organism on Earth has a functional and unique RNA sequence that makes up their ribosomal subunits. And due to their unique nature between taxa, we can use these sequences to distinguish different types of microbes. But before doing so, as groundwork for this project, we're first differentiating the ribosomal subunits of single-celled organisms, which are prokaryotes, and multicellular organisms, which are eukaryotes. And we're using an instrument called a high-pressure liquid chromatographer to do so. Essentially, prokaryotes have smaller subunits than those of eukaryotes, and we expect to see this difference in subunit size on the HPLC, which we will dig into later. So to take a step back and give you guys a broad overview of the methods that I've been a part of for this, first part of the project, is that first and foremost, we extracted RNA from model organisms. For our prokaryote, we used E. coli, and for our eukaryote, we used S. cerevisiae, which is just a type of yeast. We then moved forward once we had these extractions, and we separated the small and the large ribosomal subunits of these microorganisms on a high pressure liquid chromatographer, or the HPLC. And then eventually we're moving forward to distinguishing in a mixed media the RNA peaks of prokaryotes and eukaryotes, again on the HPLC. However, it's important to note that because this is a completely novel technique, we have to make sure that our RNA extraction quality is up to par, and to do so, we need to make sure that we have ways to quantify and qualify. So that leads into this slide here. As I mentioned, we grew the E. coli and the yeast cultures. We extracted them with these pre-made RNA mini prep kits. And then we used a nanodropper to quantify the number of nucleotides that we had from each extraction to make sure that there was enough RNA. And then also we used something called gel electrophoresis to visualize the length of these RNA strands. And that's what you guys see here in this figure. Along both sides, we have a ladder, and this is just a metric to scale the lengths of our sequences. And then we have 2 wells of E. coli samples and 2 wells of yeast. And if you look closely, you can see that we have bands, 2 bands on each organism, which is exactly what we're looking for, because these will represent our large and our small RNA subunits when we move forward and take them onto the HPLC. So once we confirmed the quality of our extractions, we were able to run E. coli on the HPLC, and here's a result that you're seeing now from using that instrument. This is an E. coli run, and as you can see on the left here, here's just a peak of single nucleotides. Essentially, RNA is a really fragile molecule and degradation happens quickly. Here's some tRNA, which is not of interest to us. And then here we see our small and our large subunits. We have some beautiful peaks, which is exactly what we're looking for. And they correlate to the small and the large subunit that's represented here by these molecules. So that was our first major success in the project, was differentiating the small and the large subunits of simply our prokaryote on the instrument. An important part of using the HPLC is creating specific buffer concentration. So here in purple, we have MPA, which is our salt solution, and then here is MPB. And this is just an example of one buffer that we use throughout the project. But essentially by using different buffers, we were able to determine it at exactly what concentrations we had our subunits eluting at. And we determined that our small subunit for E. coli eluted at 65% of MPB, and our large subunits eluted at 70% of MPB. And here's just another way to show you guys how that works. In blue and black here, we have 2 different E. coli samples run on the same, more gradual gradient, meaning that the percentage of MPB lower or raised more slowly. And we see that they eluted despite being different samples at completely the exact same time. Meanwhile, when the second sample was run on different gradients, you can see that it was eluted at different times, although at the same concentrations. And then this leads into our next step. As I mentioned, this is just the first part of an ongoing project, and there's quite a ways to go from here. First and foremost, our next step would be distinguishing prokaryotic and eukaryotic ribosomal subunits on the HPLC. Again, due to their difference in size, this should be possible in a mixed culture. Once we've narrowed down this process, we can then move forward to leveraging RNA to target finer taxa of microorganisms. And we would do this by coupling the HPLC to an isotope ratio mass spectrometer, which will give us specific isotopic values of our RNA subunits. And this ultimately is the overall goal of the project. I want to give a huge thank you to my wonderful mentors, Jim Moran and Chris Krasinski, who have been so insightful and patient and knowledgeable about of this project. It would not be the same without them. Also, thank you to the STEM staff here at PNNL, especially Nicole, Juan, and Nancy. And then lastly, to Shani Kasanke for all of her insight. It has been a wonderful experience. Thank you.
Thank you, Eve. Great presentation. Wonderful work. We can open it up for question and answer. Again, you can unmute or put it in the chat box.
Yeah, hi Eve, thanks for the presentation. I just have a quick question. So given that this was a novel technique, were there any large challenges in your project, or like in a specific example of like some troubleshooting you had to do?
Yeah, I'm glad you pointed that out. Actually, many, because this was completely new, a huge part of my experience has been trial and error, which has actually probably been my favorite part of the internship, is that we encountered tons of problems and then we got to kind of, put our feet against the wall and think about how to solve them. I would say perhaps our most recent one was finding out that using non-molecular grade salt in our buffers actually caused enough degradation in the columns of our HPLC that we had no products by the time it actually hit our column and ran on the instrument. So little things like that, just tweaks bit by bit in the process, and moving forward, hopefully things will be smoother sailing. Yeah.
Awesome, thanks, and great presentation. It was really interesting.
Thanks, Leahy.
Yeah, wonderful work. For the 10 weeks that you are here, it's or 9, I think you have one more week, but great work.
Thanks, Nicole.
Okay, thank you. Up next, again, if you have any questions for Eve, you can go ahead and message her directly. Up next, we have Claire Ma. I'd like to turn the floor over to her and go ahead and share your screen. There you go. And go ahead and unmute yourself. Thank you.
Okay, so hi all, I'm Claire Ma, and I'm an undergraduate studying computer engineering at the University of California, San Diego, currently at PNNL as a WPTO intern under mentorship by Jiaqing Yang. Today I'll be presenting on my summer project, a holistic resource assessment of offshore wind, wave, tidal current, and solar energy, a case study in Washington and Alaska. As countries begin to shift towards investing in renewable energy to replace carbon-emitting fossil fuels, much work has gone into developing marine renewable energy, abbreviated MRE, particularly in the areas of offshore wind, tidal current, and wave power. These sources of energy are promising for a variety of reasons. In the case of offshore wind power, offshore wind speeds can be much greater than onshore wind, allowing for higher power output as well as the use of shorter towers, reducing the visual impact and thus limiting the opposition from nearby communities that onshore wind often faces. Tidal currents are a constant and predictable source of power, with a global usable potential resource, taking into account tidal farm feasibility in various locations, of about 0.66 terawatts. Considering wave power, the total global wave resource has been estimated to be above 2 terawatts. As such, MRE could be a potential clean source of power for many coastal cities. However, multiple factors limit the entrance of MRE into the commercial energy market. Though offshore wind has reached commercial implementation, currently installation and maintenance costs are not yet low enough for wave and tidal energy technologies to be economically realistic. Wave, wind, and tidal resources also vary depending on geographical location. Additionally, there exists natural seasonal variability in the wave and wind resources, which may present complications when trying to produce a constant power output. These issues could be addressed in part by co-locating multiple renewable energy sources. In this way, one source's seasonal increase could make up for another's decrease, allowing for greater reliability. Furthermore, since different marine energy technologies operate in various levels above or below the ocean's surface to absorb different resources, a renewable energy farm can maximize its power output within a given area, with less interference between the devices than if using only one type of energy converter. Consequently, our project's goal was to perform a holistic assessment of multiple renewable energy resources, including the aforementioned offshore wind, tidal, and wave power, as well as solar power, at 2 locations known to have high marine resource potential. Our locations included the mouth of the Strait of Juan de Fuca in Washington, known to have high wave energy, and near the forelands of Cook Inlet, known for particularly high tidal energy. By performing this assessment, we hope to better understand whether or not sources could be co-located so as to minimize seasonal variability in power output, and the specific combination of co-located sources that would allow a renewable energy farm at these sites to be feasible. We initially estimated available power density for each source at each site, averaging over the same month over 30 years, from 1980 to 2009. These were the models and data sets we used for this estimation. For wind power density, we used wind speed data from the NCEP Climate Forecast System reanalysis dataset. Then power density can be calculated using the equation displayed. This gives us a value in watts per meter squared and gives an idea of the local resource availability before considering specific wind turbine characteristics. Tidal power density was calculated in a similar manner using predicted current speeds modeled based on the HASS 2011 tidal assessment. Wave resource can be measured using omnidirectional wave power, which we obtained from the Simulating Waves Nearshore model by Jiaoqing Yang, my mentor, and other authors. Solar power density can be quantified using downward shortwave radiation, which measures radiation energy from the sun that reaches the Earth's surface and could thus be absorbed by solar panels. This data was obtained from the NCEP CFSR dataset as well. Using these methods, we calculated a rough estimate of the resource availability of the 4 sources for a given month. We then looked at the attributes of specific wave energy converters, or WECs, solar panels, and wind and tidal turbines, choosing values that are representative of the current state of renewable energy technologies, and use that information in order to gain a better idea of the usable power output at our 2 sites. For wind turbines, we used a radius of 70 meters, which is about the average radius of current offshore wind turbines. We can then convert the wind power density into power output by multiplying by the rotor swept area, calculated using the rotor radius, and these 2 measurements of component efficiency. The power output from a tidal turbine was calculated similarly, using a radius of 10 meters instead. Although tidal current speeds can be about 1/6 that of offshore wind speeds in areas that are typically worth exploiting, the much greater density of seawater compared to air means that the tidal power density can be much greater than wind power density, allowing tidal turbines to be much smaller while theoretically producing the same power output. Estimating power output from a WEC can be done by multiplying omnidirectional wave power with capture width, a value that essentially measures the width of wave crest from which power can be completely absorbed. We used the capture width from the Polaris wave energy converter, one that was previously furthest along in development and had experienced on-site testing. In order to estimate the solar power output, we used a 96-cell solar panel made of silicon, monocrystalline photovoltaic cells. Monocrystalline cells tend to be of higher efficiency and are consequently more expensive. Since the solar resource in more northern areas such as Washington and Alaska tends to be lower, a greater efficiency solar cell may be necessary. We can estimate the power output by multiplying the power density by the total area available on the solar panel to absorb solar radiation and the efficiency of the cells. We were then able to produce estimated power output yearly trends for a given renewable energy converter at our 2 locations. Looking first at the Strait of Juan de Fuca in Washington, we see that the wave and wind power output is strongest during winter and dips somewhat in summer, with wind peaking again around June, while the solar power output is the inverse, peaking in summer. Tidal power output is relatively constant on a monthly scale. Then, looking at a few different combinations of co-located energy converters, we can estimate the potential power output and a number of households in the area that can be powered. This graph displays the monthly power output of a small hypothetical co-located farm of 1 wave energy converter, 1 wind and 1 tidal turbine, and 1,000 solar panels. The left axis displays the data and power measured in kilowatts, while the right axis displays the data in the number of households that could potentially be powered given the power output for that month. The conversion from kilowatts to number of households was based on the average Clallam County residence, which uses about 1,200 kilowatt hours per month. Clallam County was used since it is close to our case study location, so a farm at that location could potentially power nearby residences in Clallam County. Here we can see that 1,000 solar panels can account for the decline in wave and wind power output during the summer. Here is another possible combination, keeping the same number of all devices except tidal turbines, which were increased to 20. Considering the number of tidal turbines required for the power output to be comparable to wind, harnessing tidal energy in the Strait of Juan de Fuca may not be as practical. Looking at our second location at Cook Inlet, the seasonal trends in each source's power output look very similar to the Strait of Juan de Fuca, with dips in wave and wind and a peak in solar, except that wind no longer picks up in June and in fact almost disappears entirely during the summer months. When we compare the collective co-located power output for a hypothetical farm in Cook Inlet with our original farm in the Strait of Juan de Fuca, again with 1 wind and tidal turbine, 1 WEC, and 1,000 solar panels, we see that the total seasonal power output is much less consistent. Wind power output in winter and tidal throughout the year are much greater, while wave power output is practically non-existent. Again, power in kilowatts is on the left axis, number of households powered is on the right, though the conversion to number of households is now based on the average monthly energy use of an Anchorage household, about 600 kilowatt hours, half that of a Clallam County household. Due to the lower energy usage of Anchorage households and the much greater wind and tidal power output, this farm is capable of powering many more residences than the one in the Strait of Juan de Fuca. After increasing the number of solar panels to 3,000, the summer power output comes closer to that of winter. This graph also shows that the total wave power output using 100 wave energy converters is still quite low. Consequently, harnessing wave energy would not be feasible in Cook Inlet. We can then compare the average monthly power output from hybrid sources at our 2 locations. When using a comparable number of each renewable energy device, in this 1 of each except for solar panels, which is 1,000, the power output from the Strait of Juan de Fuca is mostly comprised of wave and wind power, as well as solar. The power output from Cook Inlet is comprised of wind and tidal power in addition to solar. The total power output in Cook Inlet is more than twice that of the Strait of Juan de Fuca due to Cook Inlet's much higher wind power output during winter and tidal year-round, even though the wave resource is much smaller and the solar resource is about equal. Based on our assessment of seasonal power output, a co-located renewable energy farm at the mouth of the Strait of Juan de Fuca, utilizing a ratio of about 1 WEC to 1 wind turbine to 1,000 solar panels, would be able to produce relatively constant year-round power. Due to Cook Inlet's wider disparity between wind power in winter and summer, more solar panels are necessary to make up for summer declines, with about a 1 wind turbine to 3,000 solar panel ratio. However, as Cook Inlet already possesses strong tidal resources, and tidal power does not vary seasonally, a farm sited at Cook Inlet may not benefit greatly from co-location and could instead depend entirely on tidal power. In the future, this assessment could be expanded to include a levelized cost of energy analysis to determine whether co-located farms at these locations are economically feasible. We could also examine how environmental conditions, such as weather, may impact the management of energy farms in these areas, and if cost or efficiency could be improved by the layout of the farms themselves. I want to thank my mentors and co-authors for all of the help and guidance they provided throughout my project, as well as the Water Power Technologies Office for making this project possible. Thank you for your time.
Thank you, Claire. Well done. Again, we'll open it up for a few minutes for question and answer. Please either put a question in the chat box or go ahead and unmute yourself. We have a question in the chat box, Claire. I'll go ahead and read it to you. Were there any setbacks during your research?
I don't know if there are any like specific setbacks, but I think there was a lot for me to learn about renewable energy because I was trying to consider, I guess, just like multiple aspects of implementing a farm in these 2 locations. And even though I didn't specifically cover certain aspects such as like cost and farm configuration, I still wanted to like put a lot of time into reading more about those areas so that I could like better understand the context in this assessment that I was making.
Okay. We have another question. Claire, nice work. Any idea why households in Anchorage use half the power of those in Clallam County?
Oh, that's a good question. I didn't look into that specifically, but it would be interesting to look into why that is.
Yeah, very interesting. Okay, well, Claire, well done. Thank you.
Thank
You
you.
can stop sharing. Next up, we have Jamie Oman. Jamie, if you want to go ahead and share your screen, and there you go.
All right, do you see my slides?
Yes, we do.
Hello, everyone, and welcome to my symposium presentation. Thank you for coming. My name is Jamie Oman. I'm a rising junior at University of Washington, majoring in chemistry and marine biology. My mentors are Dr. Lisa Garabelli and Michaela Freeman. Today, I will be discussing marine renewable energy in the tropics, more specifically Oaxaca, Mexico, under the direction of the Ocean Energy Systems, abbreviated OES, environmental team. You may be asking yourself, what exactly is marine renewable energy, or MRE is the acronym I'll be using from now on. Some of you might have an understanding of the subject, but for those that don't or need a refresher, MRE is a cleaner way of harnessing electricity through movement of water. It is a way for decarbonizing the atmosphere while still addressing global power demand. This energy includes tidal, wave, current, salinity, and thermal gradients, and riverine sources. At the top left is one example of a wave device positioned at the surface, which has mooring lines attached to the seabed. A point observer can generate electricity through a linear or rotary generator, or even by pumping fluid using mechanical force. The right shows a would-be underwater tidal turbine with rotating blades, which can be placed anywhere in the water column. These blades allow the turbine to change hydrodynamic operation depending on flow conditions or control settings. The bottom left is a river turbine. It can be fixed or floating, operating by harvesting energy from water that flows in one direction. With these devices comes uncertainty for the challenges associated with the environment. MRE production is slowed by environmental effects. And that brings me to this slide. So before I begin into the tropical application, I also want to give an introduction on the environmental effects of device deployment. Any proportion of an MRE device or system that might harm animals, habitats, or ecosystem processes is known as a stressor. On the receiving end, these animals, habitats, and processes affected by devices are called the receptors. It is the interaction between stressors and receptors that define the potential risks mentioned here, such as collision, underwater noise, or mooring line encounter, usually referred to as entanglement. In relation to current studies, little impact is expected from small numbers of devices, typically as before, in close proximity to one another. But larger arrays will require extensive investigation. For my project, I focused in on tropical and subtropical regions. More environmental effects are known for temperate regions such as the Pacific Northwest, but both climates have MRE potential. So why should we look at the tropics? Mexico has one of the longest coastlines and many resources go untapped. If we are able to utilize respective areas for MRE, we can increase sustainable development for Oaxaca. Considering many coastal homes go without proper electric power, installation of devices can fulfill some of the demand. At the top is a map of Oaxaca, so you can have a generalized picture of where this study is based. Below it is one of Oaxaca's popular beaches, Puerto Escondido. Over the course of my internship, I conducted a literature review in order to accumulate a knowledge base of energy in Mexico pertaining to and used for the projects of Oaxaca. I used TPIS, a PNNL platform for housing information on environmental effects of marine renewable energy, and several sites such as Science Direct and ResearchGate for critical journals and reports. In doing so, I was able to find details on the best suited resources, important socioeconomic considerations, and occurrences of marine fauna and protected habitats. This allowed me to bring together the information and connect subject relationships, such as the stressor-receptor interactions I mentioned before. Marine resources include wave, ocean currents, and thermal energy gradients, which are harvested by devices pictured here. After reading a paper describing the areas where MRE installation is most promising, I found that coastlines were some of the best choices for research. In Mexico, just offshore of Oaxaca, as circled on this resource map, there is potential for all three. Wave energy harnesses kinetic and potential energy via wave energy converters, abbreviated as WECs, which float at the water's surface. Similar to wave, ocean current devices utilize kinetic energy for water speed and flow. OTEC is a little different and uses temperature gradients, the changes in temperature between water layers, including the use of steam turbines and a thermodynamic cycle. Focusing in on the social aspect, socioeconomics, some of the biggest components of pre-installation for MRE are the people and lifestyle of Oaxaca. Cultural, industrial, natural, and public consideration is essential for proper evaluation of opinions and location selection. There are 16 main indigenous communities, including hundreds of subgroups in Oaxaca, each with its own cultural traditions and practices. Commercial, artisanal, and recreational fisheries and tourism, recreational fisheries and tourism play a role in port activity and economically favorable or dependable species with respect to spatial necessity. Marine protected areas, or MPAs, have been instigated with the intention of preserving, conserving, and restoring essential territories and resources. Practices in these areas must be managed sustainably. One MPA example is the Playa de Escobilla Sanctuary, which has restricted access during sea turtle nesting seasons. There are many other national parks and zoning programs in place to restore species in significant territories. In addition, public knowledge from previously conducted interviews suggests little understanding of what MRE is, so addressing this for future reference will be important. As you can see from this map, there are 32 recorded marine mammal species offshore within the Oaxacan coastal range. Among these include several species of whale, dolphin, and the California sea lion, each with varying migratory seasons. The olive ridley sea turtle, listed as vulnerable under the IUCN Red List, has nesting zones on the Oaxacan beaches, in addition to leatherback and green sea turtles. For diving and swimming seabirds, I found several species belonging to shearwater, tern, gull, and phalarope families, each with individuals observed and recorded offshore. Habitats are also imperative for assessment. Red, white, black, and buttonwood mangroves, as well as several coral reef ecosystems, have been notated, increasing species richness by drawing fish and more species up the trophic food web. Earlier on in this presentation, I briefly went over the stressor reflector interactions. For each type of MRE device, wave, current, and OTEC, there are specific relationships that can be applied to. The three colors represent different resources. Wave is green, current is blue, and OTEC is orange. And the table reads like a grid. 3 columns per interaction per device. Under collision, for example, OTEC and wave resources are not considered a risk, but current devices may pose a threat to marine mammals, fish, seabirds, and sea turtles from moving blades and their potential to harm or injure animals in device vicinity. Another example is underwater noise, where wave and current devices are considered a risk to marine mammals, fish, and sea turtles. Ambient noise could disrupt an animal's ability to avoid predators, communicate, navigate, interact, and forage in the environment. Entanglement may affect marine mammals and sea turtles by all 3 devices, from the use of mooring lines, cables, tethers, or anchors keeping devices in place. Oaxaca has strong potential for power sourcing of MRE devices, but careful consideration is necessary regarding the people, animals, and overall environment interaction offshore if we are to engage local communities. There is still need for additional research on the environmental effects of each device type. However, working with developers and utilizing past and current data will benefit future projects. What I have learned, communication will be key to successful MRE device deployment. More interviews and contacts with representatives from each of the community groups will provide a well-rounded opinion on the subject matter. In terms of broader scales, for example, global outreach, I've been creating an MRE brochure describing the environmental challenges of MRE project development, which can be used to engage and inform those who may not have had as much exposure to the technology. I would like to take the time to thank my mentors, Dr. Lisa Garabelli and Michaela Freeman, and the OES environmental team for all of your tremendous help with my research this summer. It has been an honor and rare privilege working with you all. I would also like to thank PNNL, the DOE Water Power Technologies Office, and the Office of STEM Education for making my internship possible. Here are my references for those of you who might need them. And on that final note, this concludes my presentation. Thank you so much for your time and attention today. Does anyone have any questions or comments?
Thank you, Jamie. Wow. Pretty interesting there. You do have a question in the chat box. I'll go ahead and read it to you. So why does tropical ecosystems have the most untapped sources of MRE? Also, great talk. You're muted, Jamie.
Sorry, that is a really great question. So a lot of the tropical regions are in more of the developing countries where certain technologies aren't as available and they're still using fossil fuels as resources. So I think that being able to communicate with them on that and being able to sort of get the locals to understand the potential benefits to MRE device deployment will help in getting the untapped resources applicable to the tropical regions, if that makes sense.
Great, great question.
Jamie, I had a quick question for you.
Sure.
So in these communities that don't have MRE at the moment, is there a specific technology that is kind of like the icebreaker of getting into renewable energies from like a cost standpoint, as well as something that appears potentially less invasive to get the communities on board?
Yes, so I would say if I could go back to my MRE table. So in terms of like an icebreaker, I would say picking certain devices that have the least amount of environmental effects, because previous interviews conducted with the locals did say that they really love their environment and the like marine fauna, so they don't want these devices to affect the wildlife. So I think that being able to determine which kind of device will have the least number of environmental effects while still giving them enough electricity to provide for their homes and communities without it could be a potential icebreaker.
Perfect. Thanks so much.
Yeah. Thank you.
Great question. Alrighty. Thank you, Jamie. We will move along. If you do have any additional comments or questions, please put them in the chat box or message Jamie directly. Next up, after this, we will have a break. So Krista Vidston, you are up next. Go ahead and unmute yourself and go ahead and share your presentation.
Hello, I am Krista Vidston. I'm going to be a senior ocean engineering student at Texas A&M. My project was gray whale detection through passive acoustic monitoring near PacWave. My mentor was Alicia Amerson, and I worked with the Triton Initiative this summer. The Triton Initiative is a project that works to reduce barriers to testing marine energy devices by researching and developing different types of environmental monitoring technologies. The Triton field trials research focuses on 4 main stressors, which are changes in habitat, underwater noise, collision risk, and electromagnetic fields. I worked on an underwater noise project to create baseline information on baleen whale vocalization activity near PacWave. PacWave is a Department of Energy funded facility based at Oregon State University that will be a future testing site for wave energy converters. This study focused on gray whales because there's been a documented reoccurring population of gray whales that inhabit the coastal waters off of Oregon, and this increases their potential risk to elevated noise levels from activities like the PacWave. The purpose of this project is to inform pre-construction habitat use and vocalization behavior of gray whales who seasonally inhabit the area near PacWave, and to help inform future researchers on potential underwater noise impacts from wave energy converters on marine mammals. 2 underwater hydrophones were deployed off the coast of Oregon from June to October of 2017 through 2020. The location of these hydrophones are shown on this map with a green M. One hydrophone was placed in the Otter Rock Marine Reserve, which is an area dominated by natural sound processes and overall has less shipping activity. The other hydrophone was deployed 17 kilometers south off of South Beach near the port of Newport. This area has a lot of ship traffic, so more vessel-related noise. These hydrophones were at a depth of around 20 meters. And on this map, the PacWave facility will be where the red star is located. The South Beach hydrophone was significantly closer to the pack wave at about 5 kilometers versus 20 kilometers for Otter Rock. This bottom diagram shows the materials and configuration of the hydrophones used in this study. Passive acoustic monitoring methods of technologies like this are non-invasive and cost-efficient ways to monitor multiple whale vocalizations in different locations over long periods of time. I used a generalized power law detection algorithm in MATLAB created by Tyler Helgley and team to detect gray whale M3 calls. M3 calls are the most common gray whale vocalization, and they have a frequency range of 20 to 200 hertz, peaking at less than 100 hertz. They typically have a duration of 1.5 to 5 seconds, and this picture shows 2 M3 calls that were found in the data. This algorithm uses parameters that the user can specify depending on the whale call being analyzed, and it applies those parameters to sound files and singles out sections of data that meet those specific requirements. I created 2 sets of parameters, and parameter 1 was used on data sets that typically had less anthropogenic noise, whereas parameter 2 was used on data sets where there was a lot of false detections with a duration less than 2 seconds. The data was whitened both in time and frequency, and an enhanced mean noise level of 3.5 was used to reduce the algorithm's sensitivity to white noise. This tool allowed for a quick and effective analysis of underwater noise data that was collected over large chunks of time in 2 very different acoustic environments. This is a spectrogram of a whale call found, and this picture was taken in Raven Pro, which is a software used to analyze sound. A spectrogram is a visual way to represent the signal strength or energy or loudness of a signal and observe how these frequencies change with time. On the y-axis here is the frequency, and on the x-axis is the duration or time. These dark blue areas are areas with low energy, and the middle of this M3 call, where it is yellow, has the highest energy. In addition to M3 calls, downsweeps were also found in this data. This is a common call produced by a wide variety of whales, such as the blue whale or the sei whale, and they have a frequency that reduces with time. This is a downsweep that has no harmonics, whereas this is a downsweep that does have harmonics, which are these extra signals right here. It is unknown exactly which baleen whale produced the downsweeps in this data. Humpback whale calls were also detected, and these whales produce a wide variety of sounds and are one of the more vocally active baleen whales. They have song calls and non-song calls, and it's predicted that in this data, there were non-song calls, which are typically described as social sounds. There were 3 main sources to false detections in this data: shipping-related noise, which was caused by hydraulics or boat propulsion; ambient noise, which was created by wind and waves; and another false detection came from crabs rubbing their shell against the pressure casing of the hydrophone, and crab activity was the largest source of false detections in this data. Looking at baleen whale calls at Otter Rock, there was a high amount of vocalization activity, with the most M3 calls being in 2019 with 82 calls. The most downsweeps were in 2017 and humpbacks in 2020. Crab activity had the highest amount of false detections, with the most being in 2018. Crabs are curious creatures, and they would inhabit these hydrophones between 2 to 48 hours. Boating-related noise was minimal, as well as ambient and unknown noises. Looking at these calls and when they were made during the summer showed some pretty cool trends. So M3 calls were most commonly found in July and August throughout all 4 years. And this is when there's typically the highest amount of vocalization activity. There were also some occasional peaks in September in 2017 and 2019. There were also some periods of low vocalization activity, and this could be because of low food sources in the area or the presence of their predator, the gray whale. More research could be done to determine if gray whales weren't in the area during these times or if they just weren't vocally active. Looking over at downsweeps, these were most commonly found in July and August, with little detections in June and September or October. On the other hand, humpback calls consistently started in the latter half of September and continuing until the hydrophone was recovered in early October. It is quite possible that the humpback calls continued past when the hydrophone was recovered in the fall months, but these vocalizations did start at the same time each year, which is pretty cool. Now comparing that to the South Beach results, there was way less vocalizations in this location, with the most M3 calls being in 2020 with only 21 calls verified. The most downsweeps were in 2017 and humpbacks in 2020. Crab activity continued to be the largest source of false detections at both locations, and there was an increase in shipping related noise, possibly due to this hydrophone's close proximity to the port of Newport, and ambient and unknown noises were also increased, and this could be due to turbulent waters because of moored vessels. So there was significantly less M3 calls at South Beach, but the time trends remained very similar. Majority of calls were found in July and August, and there were also larger periods of low vocalization activity, and these periods happened more frequently. Downsweeps were found in July and August of every year, and but 2019 and 2020 had very low downsweep activity overall. Humpback calls also started in September, continuing until early October in this location as well, and very few humpback calls were identified in 2017. So comparing the two locations, Otter Rock had significantly less anthropogenic noise due to its location in the Marine Reserve, and there was high vocalization activity and a low false detection rate. And this location was a good bit away from the pack wave, whereas South Beach was closer to the pack wave and was near the port of Newport, so it had a lot of boat traffic. There was less whale activity with higher false detection rates. So why is this study important? Baseline identification information of baleen whale vocalization levels in this area help researchers to understand prior habitat use by baleen whales of the Oregon coastal region and near pack wave, and allows for comparison post-installation and operation of these wave energy converters. This comparison also provides researchers the ability to recognize if there is a reduction in vocalization activity and if marine energy devices influence a change in behavior. During my internship, I also got the opportunity to work with the Triton communication team and learn how to effectively communicate science to different audiences. I had the opportunity to write about my story in a Triton story and about my experience working at PNNL and with the Triton team. I learned how to pitch different research projects to be featured on various podcasts and gained some cool insight on science outreach. I got to create an infographic highlighting the research that I did, and these are some data visuals that are in the infographic to be posted in Triton's newsletter. I would like to thank the DOE and the SOLI program. I'd also like to thank Alicia Amerson, Joe Haxel, Kayleen Gunn, and the Triton team for this awesome internship. And I would like to acknowledge the groups of people and programs that worked to collect this data. Thank you. And if you have any questions, feel free to ask.